Good afternoon, and welcome to today's 2024 Clinical Trial Roundup. We deeply appreciate your participation in our MSA Awareness Month programming. Your engagement contributes to our supportive and collaborative MSA community, and we look forward to continuing to create content and resources that matter to you. I'd like to take this time to thank our corporate partners that participated in our Clinical Trial Roundup: Theravance Biopharma, Alterity Therapeutics, Ono Pharmaceutical, and AskBio. We share a mutual goal with our corporate partners of pursuing improved and more accessible treatments, as well as finding a cure for MSA, and we appreciate their support of our work. Without further delay, I'm excited to present to you our 2024 Clinical Trial Roundup.
Hello, my name is Shiv Sohar. I am the Vice President of Neurology Clinical Development at Ono Pharmaceutical Company's U.S. office in Cambridge, Massachusetts. Ono was founded in Osaka, Japan in 1717, and throughout its rich history, our mission has been to develop novel therapeutics to help patients live longer, healthier lives. Ono is delving into the biology of human disease to discover new therapeutics in 4 focus areas: oncology, immunology, neurology, and specialty rare diseases. Through open innovation, leading-edge informatics, and disease modeling, we aim to advance safe and effective medicines to address unmet patient needs. At Ono, we are committed to researching neurological diseases that affect the central and peripheral nervous systems. These diseases can be devastating for patients and their caregivers. For physician scientists like me, studying and developing treatments for neurological diseases is challenging. But with the support of our team at Ono, we remain committed to discovering and developing medicines across a range of neurological conditions. Ono is committed to research that has the potential to bring patients disease-modifying therapies as well as symptomatic treatments for neurological conditions and chronic pain. Our partnerships with biopharmaceutical companies and academic institutions are helping us get closer to addressing critical unmet needs in neurology. One of our U.S. clinical studies is investigating ONO-2808, a S1P5 receptor agonist, in patients with multiple system atrophy. MSA is a rare and often devastating neurological disease that can affect motor control as well as the body's autonomic functions. At the moment, there is no FDA-approved treatment for MSA. Most therapies just manage the many symptoms patients experience. We are currently enrolling for the 2808 study, which is our first trial in patients with MSA in the United States. Please find further details about this ongoing study in MSA at clinicaltrials.gov or contact us through our website. At Ono, we are committed to working in areas where there are unmet medical needs like MSA. We want to thank our partners at the MSA Coalition for this opportunity and their collaboration. Most of all, we want to thank you for being the source of our inspiration to find medicines in this area. Thank you.
Hello, I'm Dr. Amber Van Laar, the head of Global Clinical Development at AskBio, and I'm delighted to be here to share some of our ongoing research. AskBio is a leader in the field of cell and gene therapies and is now a cornerstone of Bayer's Cell and Gene Therapy Unit. Here are my relevant disclosures. Today, we will be introducing you to the Regenerate MSA 101 trial, a phase 1 clinical trial of AB1005, a gene therapy for treatment of multiple system atrophy, or MSA. During this presentation, we will cover how this investigational gene therapy works, how the clinical trial is structured, and how to get involved if you are interested in participating in this trial. MSA is a rare, fast-progressing neurodegenerative disease that is caused by buildup of a specific protein in certain parts of the brain and spinal cord. MSA is characterized by disruption of processes that the body usually does automatically, such as controlling bladder function or regulating blood pressure. Because of this, individuals with MSA may experience bladder control issues or dizziness when standing up. MSA is categorized into two major subtypes based on the type of movement symptoms the patient experiences, in addition to the disruption of the automatic processes. Patients diagnosed with the cerebellar subtype of MSA, or MSAC, may experience a number of symptoms that result in a lack of coordination or impaired speech. Together, these symptoms are collectively known as cerebellar syndrome. Patients diagnosed with the Parkinsonian subtype of MSA, also known as MSAP, may experience the same movement symptoms as patients with Parkinson's disease, including rigid muscles and slowness of movement. Some patients with MSA have movement symptoms from both subtypes. The symptoms I mentioned are not the only symptoms a patient with MSA may experience, but they are the main symptoms used for diagnosing and categorizing MSA. The three major groups of symptoms seen in patients with MSA are related to the parts of the brain that are most affected by MSA. Disruption of automatic body processes means that neurons in the brainstem are not healthy or functioning properly. Cerebellar syndrome means that neurons in the cerebellum are under stress and not able to function. And Parkinsonian symptoms indicate neurons in parts of the brain related to movement are not functioning properly. Neurons, which are the specialized cells in the brain, are kind of like people in the sense that neurons need connection and communication to survive and thrive. The neurons in these different parts of the brain, the brainstem, cerebellum, and putamen, are connected in a large network. Dysfunction in one part can spread to other parts as the connections start to deteriorate. On the other hand, improving the health of the neurons in one area of the brain may improve the health of the connections to other parts of the brain, thereby improving the health of the system as a whole. AskBio's investigational gene therapy, AB1005, is being evaluated for patients with MSAP. So let's take a closer look at the processes behind the Parkinsonian movement symptoms. The movement symptoms in MSAP and Parkinson's disease are driven by lower levels of a chemical in the brain called dopamine. Dopamine is a key messenger in the parts of the brain that control movement, such as the putamen. In MSAP and Parkinson's disease, the neurons that control movement by using dopamine to communicate are not able to function properly and eventually die. Dopamine replacement therapy can help manage the movement symptoms of MSAP, but it does not fix the underlying disease. As MSAP progresses, the effectiveness of limited options available decreases. Currently, there is no cure or treatment to slow down MSA. Gene therapy offers the potential for treating neurodegenerative disorders such as MSA. Genes are segments of DNA that act as the blueprint for what proteins the cells in our body should make. And gene therapy harnesses this process to make proteins that can act as the medication. Gene therapy works by delivering genetic material into a patient's cells to treat the causes of a disease. There are different ways that gene therapy can work to help people, including introducing an active copy of a gene into the body to help treat a disease, or turning off a bad gene that is causing a disease, or replacing a bad gene with a healthy copy of the gene. Today, we're going to focus on gene transfer therapy, which is how AB1005 works. In gene transfer therapy, the genetic material is packaged in a carrier called a vector, which ensures the delivery to the proper cells. If you think of the genetic material as a letter, then the vector is an envelope with the address to ensure delivery to the right location. Vectors for gene therapy are commonly made from adeno-associated viruses, or AAV for short. But these viruses have been altered and will not make you sick. Scientists use viral vectors to deliver gene therapy because viruses are very good at getting inside cells. Since the genetic material of a gene therapy needs to get inside the patient's cells to be effective, viral vectors are the ideal envelope for delivering gene transfer therapy. The vector used for AB1005 is the adeno-associated virus type 2, or AAV2. So, to sum it all up, a gene, or letter, is placed in an envelope to create a package, the gene therapy, which is then delivered to where it needs to be in the body so the patient's cells can use this information to make a protein that then acts as the medication. Gene therapy could be used to preserve or restore the function of brain cells that are lost as MSA progresses. Researchers have found a loss of important growth factors needed to promote healthy function of brain cells in patients with MSA. The lack of these growth factors, such as glial cell line derived neurotrophic factor, or GDNF, may contribute to MSA disease progression. GDNF is especially important for the health of those neurons in the putamen that use dopamine to communicate to control movement. The genetic material in AB1005 is a copy of the gene to produce GDNF. AB1005 is delivered directly to the putamen through a minimally invasive, MRI-monitored, one-time brain surgery. The specific administration technique used allows the gene therapy to specifically target the structure. Any type of brain surgery is scary, but administration of AB1005 to putamen is comparatively less invasive than other neurosurgical procedures. There is no hardware left in the brain and typically no need for additional surgeries or maintenance. This is a one-time treatment that is expected to be long-lasting. Here, we have an animation showing what the administration would look like. To summarize, GDNF is important for the health of neurons that use dopamine to communicate to control movement. Loss of GDNF and deterioration of these neurons leads to the movement symptoms seen in patients with MSAP and Parkinson's disease. AB1005 is a gene transfer therapy made of an AAV2 vector that carries a copy of the gene to produce GDNF. By providing an extra copy of the GDNF gene to increase the amount of this important growth factor directly in the putamen, AB1005 may potentially support the health and survival of these neurons, which may slow down disease progression. AskBio is currently recruiting for a phase 1 clinical trial to evaluate the safety of AB1005 and better understand the biological indicators of MSAP. This study, known as Regenerate MSA 101, is a randomized, double-blind controlled trial that consists of a one-time brain surgery to administer AB1005. In this trial, participants will be randomized in a 2-to-1 ratio to receive AB1005 or a control surgery. This means that 2/3 of participants enrolled in Regenerate MSA 101 will receive AB1005, and 1/3 of participants will be in the control group. Control groups are an incredibly important part of clinical trials and are needed to evaluate if the treatment is working. Participants randomized to the control group in this trial will have the opportunity to receive AB1005 at a later date. You may be eligible for this trial if you are aged 35 to 75 years, have been diagnosed with MSAP, have the ability to walk with or without an assistive device, and have not had prior brain surgery. Enrolled participants will be required to visit the study center approximately 5 times over the first 12 months, and then twice a year for up to 3 years. During these visits to the study center, participants will undergo physical, neurological, and blood tests. In addition, participants will be asked to complete at-home questionnaires about their health and quality of life. Patients who participate in clinical trials help scientists and physicians better understand MSA and how to improve treatment. Trial participation contributes to the advancement of the medical field and may make a positive impact on the lives of people affected by MSA. For more information on Regenerate MSA 101, please contact askfirst@askbio.com or scan the QR code on the slide. Thank you for taking the time to learn about AB1005 and the Regenerate MSA 101 study. Now, I'd like to introduce my colleague, Kara, who will provide more information on AskBio and the Ask First Patient Engagement Program.
Thank you, Amber. I'm Kara Witkoff, Global Head of Patient Engagement at AskBio. Working at AskBio means working at the forefront of gene therapy. AskBio has had many firsts, including being the first to clone an AAV vector for therapeutic purposes and the first to deliver an AAV vector to the brain. AskBio is committed to exploring potentially disease-modifying gene therapies in MSA and other difficult-to-treat diseases. Across all diseases, we take pride in our patient-centric approach that focuses on collaborative patient advocacy. Ask First is our collaborative program for advocacy groups, patients, and their families who may benefit from the latest advances in gene therapy research and potential curative therapies. We welcome the voices of patients and their families to find answers for advancing therapeutics, technology, regulatory requirements, and public policy. Thank you to the MSA Coalition for including us in this event, and thank you to all who attended today. If you have any questions on what we've shared, or if you're interested in participating in Regenerate MSA 101, please contact askfirst@askbio.com or scan the QR code on the slide.
Hi, my name is David Standler, CEO of Alterity Therapeutics. I'm very happy to be participating in the clinical trial roundup and really excited to tell you about the progress we've been making over the last 12 months. At Alterity, our mission is to create an alternate future for people living with neurodegenerative diseases. We aim to modify the course of disease and hope to improve individuals' quality of life with our treatments. I'd like to summarize in the next few minutes our ongoing clinical trials in this area. The first two trials summarized on this slide are ongoing phase 2 studies in MSA. One is a double-blind trial that's called 201, and the other is an open-label trial that's focused on biomarkers, and that is also ongoing. We've recently completed an animal study investigation in Parkinson's disease with the Michael J. Fox Foundation that I'd like to share some of the key results with you. And then lastly, we have been conducting a natural history study for several years, and I'd like to show you a bit of data from that study too. Now, our target with our therapy is the increased brain iron that we see in diseases like Parkinson's disease on the left or MSA on the right. The patients are in blue. Their controls of the same age are in green, and we can see in both Parkinson's disease and then in multiple brain regions in MSA that there's elevated iron in the areas of pathology. And this is really where we're targeting our therapy. Now, our collaborators at Vanderbilt University who are participating in our phase 2 programs have been doing wonderful work in terms of measuring the iron in the brains of patients who are participating in our clinical trials. This scan that you see in the lower left-hand portion where the orange arrow is from an advanced patient. And you see at the tip of the orange arrow, that is the increased iron that is staining in the brain of this patient, that we're seeing with the MRI. And that is something that really helps us select patients for the trial, and it's something that we're trying to monitor during the course of our therapy. Now, this excess iron causes various problems within the brain. Number one, it actually promotes the clumping of this protein that you see pictured on the left. That's called alpha-synuclein, and alpha-synuclein is important for neurons to communicate with one another. So in the setting of increased iron, this protein clumps, and then neurons don't work as well. The excess iron also is the root cause for oxidative stress, which can damage intracellular structures like DNA or lipids or even mitochondria. So what we really aim to do is to reduce the iron that is in the cells, in the neurons in these areas of pathology, and to really reduce these sources of damage. And you see that pictured on the right side where the iron imbalance causes oxidative stress, the excess iron causes alpha-synuclein to accumulate. So what we really hope to do is to break this vicious cycle, preserve neurons, and improve patients' function. So how do we do this? Our lead drug is called ATH434, and this drug actually binds iron in some capacity and actually helps remove it from the brain. Now, it is an oral agent that's given twice a day, which we think is convenient for patients. And we've shown in various animal studies, but also importantly in man, that the drug does reach the site of action, which is important. And accordingly, we think it has potential to treat MSA, Parkinson's disease, and other diseases where there's increased iron. So if you want to summarize the overall strategy, our goal is to really redistribute this excess iron in the central nervous system to reduce alpha-synuclein clumping and reduce oxidative stress, thereby rescue neuronal function and overall improve, by preserving neuronal function, improve quality of life. So this is the primate study that I mentioned that was conducted in collaboration with Michael J. Fox Foundation as well as Vanderbilt University and Hamamatsu Research Facility in Japan. This is a well-established model of Parkinson's disease. And importantly, the primate is closer to humans in terms of their brain structure, in terms of their behavior. So that's why we think it's so important to validate our clinical approach. And what we showed in this study was that 434 treatment improved motor skills and general function or general behavior in these animals with experimentally induced Parkinson's disease. Importantly, the favorable impact that was observed occurred in animals with lower brain iron. So overall, we think this validates our approach towards targeting iron to treat these diseases like Parkinson's disease and MSA. So now to pivot to the clinical program, this is an image, an MRI image from a patient that participated in our natural history study. This is a 71-year-old woman with MSA. And what you see in the top are MRI images from a specialized method. And the dark red staining material is the increased iron in the brain of this individual. Now, what you see on the left, which I won't go into detail, but these are normal data. These individual dots are normal data in each of these graphs in different brain regions. And this really helps us determine where there's abnormally increased iron. So then if you try and figure out which of these units of volume in the brain on the top row actually have increased iron, we use the data from the left side of the slide to determine those voxels or tiny cubic millimeters where the iron is actually increased. And that's what we see in the bottom part of the slide. So this is something that our collaborators at Vanderbilt have been working on for several years, and we are implementing this in the ongoing phase 2 studies. So this is the natural history study I was just telling you on the left, no treatment. We're just observing patients, collecting various biomarkers and tracking their symptoms. The two trials on the right are the phase 2 studies that I mentioned at the beginning. The 201 study in the middle column is a randomized double-blind placebo-controlled study. The one on the right, the 202 study, is our open-label study. Both are looking at the efficacy and safety of 434, slightly different populations, and we are treating patients and following them for 12 months with a focus on brain iron, on MRI, but importantly on clinical activities of daily living, autonomic function, and various other parameters. So what I've been telling you about to date is really on the left side of the slide, what we kind of completed at the end of 2023 and early 24. Looking forward to later this year, we hope to have preliminary data from the 202 open label study by the June timeframe. And then we will complete treatment in the 201 study in November of this year with having data from that study by the end of the year or by early next year. Finally, we will be presenting some new data from our various studies at the American Academy of Neurology in April. So with that, I'd like to conclude and thank you for your attention and hope you have a great conference.
Good morning. My name is Rick Winningham, and I'm the Chief Executive Officer at Theravance Biopharma. It's my pleasure to present the Cypress trial, a new clinical trial that our company has initiated that will study the investigational drug known as ampreloxetine for the treatment of neurogenic orthostatic hypotension in patients with multiple systems atrophy. This is a very important program to me. My father had multiple systems atrophy and had terrible neurogenic orthostatic hypotension. To be able to make a difference in this disease, to help the patients, to help caregivers with a potential new medicine is one of the most meaningful projects that I've undertaken in my career. The Cypress study is a global clinical trial that Theravance is currently enrolling for participants with MSA. The phase 3 trial will last for 20 weeks and test whether our investigational drug, ampreloxetine, exhibits benefits for participants that have neurogenic orthostatic hypotension and MSA. The study will also examine whether ampreloxetine is safe and will learn about any side effects. Participants that complete the 20-week portion of the study will then be eligible for entrance into a long-term extension in which all patients will receive ampreloxetine for 2 additional years so that we can study the long-term effects of ampreloxetine. We're excited to make you aware of this particular study, and we'll tell you more about it during our presentation today. Ampreloxetine is an inhibitor of the reuptake of a neurotransmitter called norepinephrine in the nervous system. We're studying ampreloxetine in a phase 3 trial, which is the final stage of drug development prior to submitting a drug to the FDA for approval. It's important to recognize that ampreloxetine is still being investigated and is not approved by FDA for the treatment of NOH. The administration of ampreloxetine is convenient. It's given once a day orally in a small tablet. Ampreloxetine's also been studied in over 800 clinical trial participants to date in various disorders, including multiple systems atrophy. So how does ampreloxetine work? The top half of the slide depicts the situation in which people have neurogenic orthostatic hypotension and MSA. Under these conditions, there is insufficient norepinephrine released when a patient stands up. Therefore, upon standing, the blood pressure will fall because blood vessels do not constrict as they would following the release of normal levels of norepinephrine. Often, this drop in blood pressure will result in the symptoms of neurogenic orthostatic hypotension, including dizziness, lightheadedness, as well as other symptoms such as vision impairment, weakness, fatigue, trouble concentrating, and head and neck discomfort or coat hanger pain. Ampreloxetine blocks the norepinephrine transporter and therefore increases the levels of norepinephrine present when you sit or stand up. The increasing level of norepinephrine should result in a relative increase in blood pressure. We hypothesize that this increase in blood pressure can also increase blood flow in the brain, which may help reduce the symptoms of neurogenic orthostatic hypotension, such as dizziness and lightheadedness. Ampreloxetine was studied in a prior clinical trial, Study 170, and demonstrated a benefit in both standing blood pressure and NOH symptoms in the 40 combined MSA participants that were treated with ampreloxetine when compared to a placebo pill. The benefit with respect to standing blood pressure is shown here, and those that received ampreloxetine had a systolic blood pressure upon standing that was approximately 18 millimeters of mercury higher than those who had received a placebo pill. Importantly, the benefits that were observed on blood pressure were also seen when symptoms of NOH were evaluated. As you can see here, the symptoms of ampreloxetine were observed across a constellation of symptoms of NOH, including dizziness, vision impairment, weakness, fatigue, trouble concentrating, and head and neck discomfort. The magnitude of these changes was clinically meaningful, and the effect was maintained over a length of 22 weeks. In addition, To the favorable impact on symptom score, there was also an improvement in daily activities that required walking and standing for a short time. Theravance Biopharma is now interested in confirming whether ampreloxetine is effective and safe in treating NOH symptoms in MSA patients and to learn about any unwanted side effects. Ampreloxetine was generally well tolerated in the previous trial, with the most commonly reported adverse events being headache and urinary tract infection. The phase 3 Cypress trial has been designed to test the durability of effect of ampreloxetine over 20 weeks in participants with MSA and neurogenic orthostatic hypotension. All participants will receive ampreloxetine for 12 weeks during an open label period, followed by randomization to either placebo or ampreloxetine in a blinded fashion during the randomized withdrawal period for an additional 8 weeks. A composite assessment of the symptoms of neurogenic orthostatic hypotension, which I've reviewed before, including dizziness, vision impairment, weakness, fatigue, trouble concentrating, and head and neck discomfort, will form the basis for the primary evaluation and results for this study. All patients that complete the initial 20 weeks of the trial will be invited to continue participating in a long-term extension and receive ampreloxetine in an open label fashion for an additional 2 years. You may be eligible to participate in the Cypress trial if you meet several key criteria. 1, if you've been diagnosed with multiple systems atrophy. 2, you have neurogenic orthostatic hypotension as demonstrated by reductions in blood pressure upon standing. And 3, you have symptoms of dizziness associated with your orthostatic hypotension. The Cypress trial has been designed to help minimize some of the challenges encountered during the clinical trial participation. For example, to alleviate the burden of travel, all visits except the first may be conducted in either the clinic or hospital setting or at your home. You may be wondering how you can participate in the Cypress trial. We're currently opening study centers across the United States, Canada, and worldwide. For more information regarding trial participation, including the locations of study centers, please email us at cypress@theravance.com, give us a phone call at 1-855-633-8479, or visit our study website at www.cypressstudy.com. Theravance Biopharma is committed to improving the lives of MSA patients with symptomatic NOH. Please consider joining us as we take another critical step toward the goal of managing symptoms of neurogenic orthostatic hypotension for people with MSA. Thank you very much for your attention and your interest in clinical trial participation.